Well, good evening, Mr. and people who are watching us. In this opportunity, we're going to talk about the second practice of a laboratory of food microbiology. In this case, we're going to talk about the calculation of G value. What is this G value? This G value is the generation time, which is going to be calculated with the absorbance of the bacteria E. coli throughout the time. So let's start. We're going to first introduce something about what we're going to do. Then we're going to say all of the steps that we took to do this practice. Then we're going to talk a little about the graph that we obtained in this experiment. And then we have some final questions that the teacher asked us to answer. And finally, a little conclusion about all of the things that we wanted to talk about. So let's begin. Firstly, what is this practice about? Well, our aim, as I mentioned before, is to calculate the G time, the generation time. To do this, we have to measure the absorbance of the E. coli. This E. coli that we're going to calculate is going to be in a solution. So this is going to be in a solution that we're going to take some volume of it and going to calculate the absorbance in two specific measurements. The first in 500 nanometers and the second in 600 nanometers. Well, actually here was 550 nanometers. What is this generation time? The generation time is how much it takes to duplicate the population of a bacteria. So it means that, for example, if I had 4 bacteria, how much time will it take to have 8, for example? Now my partner is going to talk about the methodology.
In this case, I will talk about the methodology about this practice. In this practice, we divided by different parts. In this case, 4 parts about this methodology. The first part is preparation of microbial cultures. In this case, the microbial strain of E. coli was inoculated into the test tube or Erlenmeyer flask containing the appropriate liquid culture medium. Something important to mention is that for the sake of time, the laboratory technician is in charge of solving test tubes every hour, so that we can concentrate of analyzing the absorbency in the practice. The second part of this process of the growing on the E. coli is that microbiology ground measurement. In this case, using 3 different parts about this process, such as use gloves, lab coat, and masks. The letter C is take aliquots of microbial culture at the regular intervals every 5 minutes during the exponential growth period. And letter D, measure the absorbency of each aliquot using a spectrophotometer at a wavelength of 550 nanometers and 600 nanometers to estimate cell density. In this slide, you can see some examples of the process of obtaining absorbency at 550 nanometers and 600 nanometers. This picture is encouraging made samples because in this practice, we have a little file about this. And the letter C about the process of the practice is divided by 2 practices. The practice number 1, the microbial ground data. In this case, the plot of the absorbency of E. coli in 550 nanometers and 600 nanometers versus time. You can see in this picture different types of absorbency of each group in this class, and divided different parts of the absorbency and the groups and the incubation time and mediation time. And finally, the practice number 2 is the microbiology ground data graph and the plot of the absorbency of the E. coli on only the 500 50 nanometers as a function of the time waited. In this case, the practice 2 is so different to the practice 1 because in this practice, the graphic is not coherent. In this case, the practice 2 is so coherent to express in the graph. You can say this time waited and the absorbancy coherence about to graphics in app of the graphics and the is absorbancy of this practice of a spectrophotometer.
Well, continuing with the analysis of David, we saw that we made two graphs to compare the growth of these microorganisms. You know that for any microorganism, specifically bacteria, what we hope to have, or like what we are waiting to have, is to have like exponential growth. However, as you can see in the first photograph, or the first graph on the left, we can see that, well, this is like sort of a mess. You know, like all of these data are not well organized. Well, here we had in just one graph two specific measurements: 550 nanometers and 600 nanometers. The former are going to be the ones that are going to be in color blue and red. It means that going to be the 550 nanometers are all of those points. And the second, the 600 nanometers, is going to be the ones that are just in color blue. You saw here like something that is not very well organized, as you can see, for instance, in 600 nanometers, that we started with a growth. As you can see here, there is a growth, and there it keeps growing and growing. However, then it starts to go down again. You see, it goes down, and then it goes up, and then goes down once again. So it means that this doesn't make any sense compared with what we hope to have, like exponential growth. And the same happens in 550 nanometers. We started here, it started growing, but then it goes down, it goes even much down again, up, down, and then it goes up again. So this is like not so coherent. On the other hand, and as our partner told us, is that this time is much more coherent. As you can see here, we have like a growth. Of course, that is not exponential because not all curves are perfect. However, it has like much more sense because as we have here like the time weighted and absorbance, that what we did here is that every 10 minutes, what we did is to take an aliput of the E. coli solution and to put in the incubator. We increase the temperature, and there we saw like a growth in the microorganisms. We're going to talk about temperature in just in the final questions, but the temperature what did is to increase the growth of the microorganisms, and there we saw there is a growth and the absorbance increase as well. So that is what we had here in this example. Well, the next part, here we have like the calculus of the generation time. What is it about? We said that it's going to be the time that it takes for a bacteria to duplicate itself. To do this, the only thing that you have to do, in this case, is to subtract T2 minus T1 and to get the magnitude of it. Yeah, so in this case, we just have 10 minutes per each because we took the example every 10 minutes. Then we have to take the logarithm 2 n2 divided by n1. Then there we have this. Then we have these numbers, and finally the g time is just the division between T2 minus T1 and the second data. So there we have these examples. You might be wondering, are these numbers the proper time? Well, to know it, you can see this final part, which is the one that we couldn't calculate. But why? The thing is that, as you can remember here, in this specific part, we stopped having a result. It means that the bacteria stopped growing, and that's actually what is happening here. It stopped dividing itself, and therefore is that we don't have any number here. The rest of the numbers all are also coherent, and it's more important to remember that we have here in minutes. Yeah, because the time here is in minutes. Also, the final result of t is gonna be minutes. Let's move ahead. Here we have the final results. I think that I already gave you some of these final results that we had, but to summarize or to give you um, like summary of what we've done is that the first part, the first experiment, the first practice were very good results. We didn't have very good results because there were some problems. We speculate that the problem that we had there is that maybe is in I believe that is in the when we measure the absorbance, because to measure the absorbance we have to have like, firstly, we have like sort of a cube, and this cube has to then have like a sort of an arrow, so the arrow has to be like in front of the light. Maybe that was a problem, that we didn't put accurately in the tube in the absorbance equipment. That would be one reason. The second reason could be, and I hope to be mistaken, is that the assistant, the technician assistant, did something wrong in the experiment. Maybe this person put more of the solution. It could have been that, but actually we have been like thinking about it. So, and our teacher told us that the problem could have been that we put too much, so we put too much bacteria inside the solution, and therefore we couldn't calculate very well the absorbance, because as we had more bacteria there, it was that in the light couldn't pass very well, or something related with that. And yeah, so now we're going to talk about the conclusions, which is in charge of my partner.
Well, finally, I will talk about the conclusions about this practice. The fair practice of the absorbency was not accurate, so it was repetitive again with the E. coli at 600 nanometers and using the incubation every 5 minutes. This time there was more attention to apply the form, procedure of the inputs and equipment correctly. And about the practice two is that our second data collection was successful, although almost the same procedure as the practice one. Wasn't applied, but a more coherent curve that the previous one could be obtained. This is not perfect, but is too similar to growing of the E. coli bacteria.
Well, and final questions, as I told you before, we have these questions. The first one says, why is it important to know the generation time of microorganisms in food microbiology? Well, I think that even though I have said this many times before, it is just important because you know the time that the bacteria is going to duplicate. So imagine that any bacteria, for example, Lactobacillus GG, you have like milk, and you want to like make some yogurt. So it's going to be important to know how in what time the bacteria is going to duplicate. Because if we have a lot of bacteria in the yogurt, a lot of lactobacillus, it could be like something dangerous for people who are going to consume these products. Well, to moving ahead to the second question, we have, how do environmental conditions such as temperature, pH, nutrient concentration influence the generation time of microorganisms in food? Well, I always say that every species or being on Earth has some specific characteristic or features to have to grow. For instance, humans, they have to be in a temperature, a temperature between, I don't know, let's say between 0 to 40 degrees. If we are out of that interval, it's much more difficult to live. It's the same in bacteria. If, for example, you change the conditions of bacteria, they're not going to live well, because inside them, in their structure, their molecules, their proteins, their lipids, their phospholipids, all of these things that the bacteria is made of, are going to suffer because they are not accustomed to, for example, to a specific temperature or to a specific pH. To give you, well, a case in point is E. coli. For instance, in E. coli, we know that this is a mesophil. This mesophil E. coli, it can only live between I don't know, 20 Celsius, grade Celsius to 4 grade Celsius. So if we pass that range, it's like this microorganism cannot survive because it's not accustomed to those to other temperature. The same in pH. This microorganism, well, a facultative aerobius, has a pH of 7. So if we lower the pH, it's gonna be so acid. There's gonna be a lot of H3O plus inside it, so it's gonna be much more difficult for them to do. So, well, in the same with nutrients. Imagine ourselves, humans, if we don't have meat, poultry, all of those foods, we are not gonna live well. It's the same for microorganisms. If they don't have like if they don't have the food that they can, so that they can survive, they're gonna die. That's all. That's all.
The third question is, what relationship exists between generation time and the exponential growth phase in the growth curve? The relationship about this is that the generation time is related to the exponential growth phase, represented in a growth curve. In this phase, microorganisms multiply rapidly, leading to a rapid increase in population size. Specifically, generation time denotes the time required for the population to double during the exponential growth phase. The fourth question is, what is the importance of performing absorbance measurements at 550 nanometers to estimate cell density in microbial cultures? In this case, exists for three different reasons. That is a colligram characteristics, optical wavelength, and calibration and comparison. In this case, is commonly uses a model organisms and is suitable in estimating cell density of each microorganisms, E. coli. And too sensitive to change in cell density and minimizations of interference of medium components. And the cell density facilitating occurrence, qualifications, and comparison of cell densities. And the last question is, what other methods can be used to measure microbial growth and calculate generation time? In this case, a variety of methods are available to predict cell growth by direct or indirect measurements, such as cell dry weight, cell optical density, cell turbidity, and cell respiration. Metabolic rate and metabolites as quite suitable for analyzing cell growth, substrate utilization of products information. These are different methods that we can use to measure microbiology and calculate the generation time of these microorganisms calling E. coli.
So, well, that's all for now. Thank you for watching this video, Mister, and people who also are here. So, we will be creating more videos on future dates. So, thank you. Bye.